who holds a master's degree in pharmacy, with a focus on the pharmaceutical industry. And she currently working a PhD at Sciensano at the University of Belgium on a research project investigation of resistance mechanisms and their transmission in emerging pathogens applying the One Health concept. On va parler, je pense, de Klebsiella pneumoniae et Aspergillus fluviatus.
I will do it in English. Today it's only about Klebsiella pneumoniae. I only have 10 minutes, so I cannot talk about both. So today I will talk about Ertapenem resistance in a Klebsiella pneumoniae strain that we isolated from a dog, so a companion animal, and I will present a case study here. So Klebsiella pneumoniae has been mentioned before today. It is a gram-negative bacterium, and it can act as an opportunistic pathogen. It is commonly in the environment, it is commonly found in wastewater and so on, and as well, as mentioned before, in human and animal microbiota. It can cause, among others, pneumonia, urinary tract infections, sepsis, wound infections, and so on. You all know it by now because almost everybody said it. The ESBL producing and Permeability resistant strains of Klebsiella pneumoniae are listed in the WHO list of urgent threats for which new antibiotics needs to be developed. Klebsiella pneumoniae has also been called the canary in the coal mine because some of those well-known resistance genes have been first detected in Klebsiella pneumoniae, as it is also a very well-known trafficker of antibiotic resistance genes. So it can take up really easily those resistant genes, but it can also distribute them quite easily. The EMA has developed a certain guideline for categorization of antibiotics for use in animals, where they adopt the One Health approach, so where they look at the interaction between animals, humans, and the environment, for which in Klebsiella it's quite important. As I said, it's quite prevalent in the environment, but also in humans and animals. In this categorization, they make use of 4 categories. So the first one is antibiotics that should be avoided in veterinary clinics. However, you can see it in the third point in category A that it can be given to companion animals in exceptional cases. In category B, we have the antibiotics that should be restricted. Category C is those that should be used with caution, and then category D is those that should be used with prudence. If we look at category A, then you see the carbapenems are in this category. So here, meropenem and doripenem are listed. Then if we look at carbapenem resistance mechanisms, there are 3 main mechanisms which can lead to carbapenem resistance. So the most known one is the presence of carbapenemases, but also you have pore mutations which lead to permeability defects, and then also increased efflux pumps. If we look at the carbapenemases, there are 4 classes, so class A, B, C, and D. For example, in class A you have the well-known one KPC, which is often seen in human cases. And then also, for example, in class D you have the OXA, so the OXA48 and OXY48 likes. As mentioned here, I will present a case study. So the case study is a 4-month-old female dog, a Rottweiler, that died in June 2020 due to fatal sepsis. Post-mortem biopsies were performed by Garcia of the liver and brain. Sadly, we did not receive any information about possible treatments that the dog received. This study was published in Antibiotics, so if you want more details, you can go there. So as mentioned, the dog died. They brought it to Garcia. A liver biopsy and a brain biopsy was performed. And they used Gassner and Colombian agar to isolate presumptive Klebsiella pneumoniae cases. The strain was sent to Sciensano, where I performed identification using multi-locus MS. Then it was detected or identified as presumptive Klebsiella pneumoniae. I say presumptive because multi-locus is not always very correct. It can give wrong identifications because of the database that they have. Then antimicrobial resistance testing was performed, and here we saw that there was it was resistant to cefotaxime and so on, so we performed also the second panel. And then afterwards we performed hybrid sequencing, so using Illumina and nanopore technologies so that we could circularize the plasmids. So first we performed the first panel, so of set 3, where we saw that it was resistant to cefotaxime and ceftazidime, indicating that it was an ESBL. But then again, to go more into detail to really identify which type of ESBL, was it maybe AMP-C or so on, we performed the second panel where we saw that it had an ESBL and AMP-C phenotype, which you can see because it has cefotaxime resistance, ceftazidime, cefoxetine resistance, and then also a synergy between cefotaxime, ceftazidime, and the clavulanic acid. But here, for us, the importance was also that it was resistant to Ertapenem, but sensitive to imipenem and meropenem. If we see a resistance to Ertapenem, imipenem, and meropenem, it is indicative for example, an OXA48 or something, but here we only saw the Ertapenem resistance. So that's why we went on with the sequencing to look at the resistance mechanism. Sequencing revealed that it was an ST11, and ST11 is part of the clonal group 258, which is a known global problem clone, so to say. So they are widespread all over the world, and often they are MDR clones. Also, ST11 is usually a human clone, so it's usually seen in human cases. And as mentioned before, often carbapenem-resistant isolates in companion animals is related to humans. It is not part of this study, but we are also doing a study where we sequenced over 300 isolates coming from the food chain, wastewater, humans and animals, so diseased animals. And by chance, actually, we saw a group of isolates that only differed by 11 SNPs from, so two human isolates only differed by 11 SNPs with these isolates, indicative of a clonal spread, but we don't have any more information. When we used the nanopore technologies, we were able to circularize the plasmids. We identified 3 plasmids, so the INCF1B, the INC-R, and the CoRNA-A. In the INCF1B plasmid, we saw the presence of beta-lactams, BLA SCO1, BLA-TEM1, and SHV2. SHV2 is an ESBL gene, and then SCO1 has weak carbapenemase activity. In the INC-R plasmid, we observed the DHA1 gene, which is a AMP-C gene, which is also related to the AMP-C phenotype. And then the third plasmid did not have any antibiotic resistance genes. And then on the chromosome, we observed the outer membrane pore in OMCA37, which, if there are some mutations there, could lead to porin deficiencies. So the first plasmid is the INCF1B, where we located the BLA SCO1 on the bottom there. And then also the transfer genes, so meaning that this plasmid is a conjugative plasmid, so it can be transferred to other Klebsiella species or even other bacterial species, which is worrisome. On the second plasmid, we observed the DHA1. This is, however, not a conjugative plasmid, but again, the presence of those both plasmids in the bacterium did eventually cause the Ertapenem resistance, alongside with the mutations inside the OMCA37, because then it led to pore deficiencies. So one thing here I would like to highlight is that the use of whole genome sequencing in those cases is quite important. You may say, yeah, but it's only one case. But in general, we usually look only at those strains that are Meropenem, Imipenem, and Ertapenem resistant. But here, yeah, it's important to also look at the other types of resistance, because, for example, in Klebsiella, it's quite often that we only see Ertapenem resistance, whereas in E. coli, it's not very often, actually. So we described here the fatal sepsis of a 4-month-old Rottweiler that had the MC and ESBL phenotype, together with Ertapenem resistance. We did not find any carbapenemase genes. However, on the 3 plasmids that we identified, we observed the SCU1 and the DHA1 together with the permeability defects, which in this case led to Ertapenem resistance. Thank you.